Trilata organic farming. We will see the microbes permitted in organic farming. Viral preparations like granulosis, nuclear polyhedrosis virus like Helicoverpa and Spodoptera and PV. These things are permitted in organic farming. Fungal preparations like Trichoderma, which is the fungus which controls the fungal diseases, also permitted. Bacterial preparations are permitted. Parasites, predators, and sterilized insects, male sterile insects, and all are permitted. Other category ones which are permitted in organic farming: carbon dioxide and nitrogen gas restricted to use to control pests and diseases. Soft soap, potassium soap permitted because soft soap is permitted to use along with the microbial preparations and other botanical sprays. Ethyl alcohol prohibited. In any case, ethyl alcohol should not be used in organic farming. Homeopathic and Ayurvedic preparations permitted. Herbal and biodynamic preparations permitted, like a biodynamic tree paste and all, whatever biodynamic formulations are there, all permitted. Traps, when it comes to the trap, physical methods of a control like chromatic traps, mechanical traps, sticky traps, and pheromones, all are permitted. The microbial preparations, like viral preparations, mostly in India, uh, two NPVs, Helicoverpa NPV and Spodoptera NPV, are very popular. This entire content, how to prepare Spodoptera NPV and Helicoverpa NPV, I will explain in separate session as complete PPT. Here just I am telling you that it is permitted. Both the Helicoverpa NPV and Spodoptera NPV are permitted. To produce NPV, first, if it is Spodoptera NPV, Spodoptera litura is to be produced. The larvae are to be produced. That means entire insect is to be cultured. To produce Helicoverpa armigera, Helicoverpa armigera is to be raised first. The larvae, host larvae is to be raised. Then they are to be fed with the NPVs. NPVs are very, very specific. Spodoptera NPV will not work for any other pest, including, suppose if it is Spodoptera litura NPV, it will not work for even Spodoptera frugiperta. It will not work for Helicoverpa armigera. The same way, Helicoverpa armigera NPV will not work for Spodoptera litura or Spodoptera frugiperta or any other insect. So, highly specific. The NPV works only for that insect. That's why host insect is to be raised first. Then it is to be used to control the specific pest in the field. Fungal preparations, there are various types of fungal preparations. Most important one is Trichoderma. Trichoderma is a fungus which controls the other fungi. I will explain the production of Trichoderma, on-farm production also, if how farmers or anyone can produce the Trichoderma in their own farms in a separate PPT form. So, entomopathogenic fungi, these are the fungi which controls the insects. Trichoderma is a fungus which controls the other fungal diseases. Entomopathogenic fungi are the fungi which controls the insects. Most important entomopathogenic fungi are Bavaria bassiana. Actually, this Bavaria bassiana is very wide spectrum one. It can control thrips, whiteflies, aphids, termites, ants, beetles, weevils, grasshoppers, mealybugs, and caterpillars. Then Metarhizium anisopliae. This is also very effective against fall armyworm, weevils, grasshoppers, and beetles. Nomoria rilei, it is a little bit difficult to produce Nomoria rilei. Otherwise, it is very good fungus which causes diseases to Spodoptera frugiperda, Spodoptera litura, Helicoverpa armigera, Trichoplusia knee, Anticacia, and Pseudoplusia. So, it is effective on many insects. The next fungus, entomopathogenic fungus, is Lecanicilium lecani. Earlier, it was called as Verticillium lecani. That's why it is popular as Verticillium lecani only. Many people calls now also it as Verticillium lecani only. It is also known as white hollow fungus. This Verticillium lecani is called a farmer's friend because it was having wide host range of insects from orders Diptera, Hymenoptera, Lepidoptera and sucking pests such as coccids, aphids, thrips, whiteflies and mites. The fungus is also effective to several species of nematodes. Verticillium lacani, Metarhizium anisopliae, Pseudomonas fluorescens, Trichoderma horsianum, Trichoderma viride, all these helps in controlling the nematodes. Escherichia species, earlier Escherichia farinosa and Escherichia fumazorosea were called as Bacillomyces farinosus and Bacillomyces fumosorosis respectively. Both fungi are found throughout the world and possess a broad host range. Actually, Escherichia farinosa is of less importance as biological control agent, whereas Escherichia fumazorosea is considered as a species complex, and different strains are successfully used for control of nymphs and adults of whiteflies and several insect pests. This causes yellow muscardine disease, and it is one of the most important natural biocontrol agent. It causes strong epizootics, especially in whiteflies, Bemisia and Trialurodes. Even other whiteflies are also susceptible to Escherichia uh, in both greenhouse and open field conditions. Hirsutella. Hirsutella also consists of more than 50 species of entomopathogenic fungi, but only few have been reported as pathogenic to Eriophyid mites. The three important species of the genus Hirsutella are Hirsutella thomsoni, Hirsutella gigantea, and Hirsutella citriformice. Hirsutella thomsoni is used for control of citrus rust mite, Phyllocopriola olivorus, and it is also pathogenic to Acarida, Lepidoptera, and Hemiptera insects. Nowadays, Hirsutella is also gaining importance in chili to control the chili pests like thrips. So these entomopathogenic fungi, how to produce entomopathogenic fungi on farm production, I will explain through APPT in detail. So in our next session, let us see bacterial preparations which are permitted in the organic farming. Thank you. Signing off, Srilata.